Now, I discuss about the meiosis cell division because mitosis cell division you also did in class 10. For that, now I discuss about the meiosis cell division. In meiosis cell division, four daughter cells will form a single mother cell, and each daughter cell contains half of the chromosome. For that, the meiosis cell division. The meiosis cell division is also called reductional division. It occurs in our reproductive cell. Means it occurs in gamete mother cell. Okay, at the time of gamete formation. Sometimes it also occurs in the spore mother cell at the time of spore formation. Okay, so the meiosis cell division separate the we can separate the meiosis cell division into two phases. These are the meiosis 1 and meiosis 2. It occurs by two steps. First step is meiosis 1 and another is meiosis 2. Meiosis 1 also classified into two phases. These are the karyokinesis 1 and cytokinesis 1. Then the karyokinesis 1 also separated into four phases. These are prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1, metaphase 1, anaphase 1 and telophase 1. Prophase 1 of the meiosis 1 also separated into five subphases. That is the most important part of the meiosis cell division. These are the leptotin, zygotin, pachytin, diplotin and diakinesis. Remember it, the four subphases of the prophase 1 means leptotin, zygotin, pachytin and diplotin occur inside the nucleus of a cell. After the diplotin means at the time of diakinesis, the nuclear membrane and nucleolus will disappear. The nuclear membrane and nucleolus will disappear, and all chromosomes are comes into the cytoplasm. So first the subphase is leptotin. In leptotin the chromosomes are formed from the chromatin fiber of a cell. The chromosomes will form from the chromatin fiber of a cell. Then the zygotin. In zygotin, two homologous chromosome will pair. Means diploid cell. In a diploid cell means twin cell, the cell contains another copy of each chromosome, and those another copy of each chromosome is called homologous chromosome. We can also define the homologous chromosome as the chromosomes which have same size, same shape and also same gene arrangementation is called homologous chromosome. And the homologous chromosome come closer to each other and paired with the help of synaptonymal complex. And the pairing of two homologous chromosome is also called synapsis. The process of pairing of two homologous chromosomes is called synapsis. And in synapsis, a special protein takes part is called synaptonymal complex. By the help of synaptonymal complex, two homologous chromosomes are or will paired. Then the pachytin. In pachytin, each chromosome of the homologous pair longitudinally divided without their centromere region. For that, each chromosome contains for that at that stage means at pachytin stage, each chromosome contains two chromatid. Means in a homologous pair, the chromosomes contains spore chromatid. For that, this stage is called tetrad or tetravalent. Okay. Then in packaging, at the time of dividing of the chromosomes, two non-sister chromatids are crossed in a point and form a X-shaped structure This X-shaped structure is called chiasma or chiasmata. This X-shaped structure is called chiasma or chiasmata. The next part of the chiasma will interchange in between the non-sister chromatid of homologous pair. This process is called crossing over. This process is called crossing over. So the interchange of the part or body of a between non-sister chromatid of homologous pair is called crossing over. After crossing over, each chromosome gets a new gene arrangement. After crossing over, each chromosome gets a new genetic arrangement. It is called recombination. And in that case, or in the case of crossing over, two enzymes will take part. These are nuclease enzyme, ligase enzyme. These are nuclease enzyme and ligase enzyme. The nuclease enzyme cuts the chromosomal part. The nuclease enzyme cuts the chromosomal part from the chiasma. Okay, and interchange will occur in between two non-sister chromatid, and then ligase enzyme helps to attach with the chromosomal parts. Okay, and another enzyme named as the recombinase enzyme. Recombinase enzyme helps in the recombination of each chromosome. Then diplotin. At diplotin stage, the synaptonymal complex will break down. For that, the homologous chromosome will separate it. For that, or at the time of separation of homologous chromosome, the chiasma. If it will the chiasma and it is the synaptonymal complex, then synaptonymal complex is breakdown. For that, the homologous chromosomes are separated. For that, the chiasma moves to the end part of the chromosome. This process is called terminalization. So terminalization is occurred at the diplotin stage. And then the last stage of the prophase 1 is diakinesis. In diakinesis or at diakinesis phase, the homologous chromosome will completely separated. And nuclear membrane and also the nucleolus also disappear. Okay, and nucleolus also disappear. And all chromosomes which are present inside a nucleus will comes to the cytoplasm of the cell. Then the metaphase. In metaphase, all chromosomes are arranged in equatorial plane. Then anaphase, by the contraction of the spindle fiber, the homologous chromosome moves to the pole of the shell. And then telophase 1, when the homologous chromosomes are getting into the pole, then nuclear membrane and nucleolus reappear and new nucleuses are formed. Look at that stage. In telophase 1, a single cell contains two nucleus. Then cytokinesis 1. After completing of the karyokinesis 1, cytokinesis 1 will occur. In case of animal cell, a furrow will form at the middle portion of the cell. A furrow will form at the middle portion of the cell, and this furrow are moved or elongated inwardly. When they completely attached, then two daughter cells are produced. Then the meiosis 2 will occur. In meiosis 2, first phase is karyokinesis 2. Meiosis 2, the steps of meiosis 2 same as mitosis cell division. In prophase 2, the nuclear membrane will disappear, nucleolus also disappear, and the centrioles will divide and move to the pole of the cell and moves to the pole. Okay. In metaphase 2, spindle fiber will form, and the chromosomes will arranged in the equatorial plane of the cell, and spindle fiber gets attached with the centromere of each chromosome. Then the division of centromere also occur. For that, each homologous chromosome means two chromatid of each chromosome will separate it. Then anaphase 2. In anaphase 2, by contraction of the spindle fiber, each chromatid of the chromosome moves to the pole. In telophase 2, the chromosomes are, when the chromatids are completely reached at the pole, then nuclear membrane also reappear, nucleolus reappear, and new nucleus will form inside the cell. Look here, at that state means telophase 2, a cell contains two nucleus, and then after completing of the karyokinesis means nuclear division, cytokinesis 2 will occur. In cytokinesis 2, here a furrow will form in between. A furrow will form at the middle portion of the cell, and the furrow elongates inwardly. When they attached, then two daughter cells are formed from each cells. For that, after completing of meiosis, we can get 4 daughter cells from a single mother cell, and the chromosome number will decreases after the meiosis 1. For that meiosis 1 is the most important part for meiosis cell division, because in meiosis 1 the chromosome number of a cell become half. Then why does the meiosis 2 will occur? Meiosis 2 will occur for the distribution of the chromosomes, in the cells. Okay. Remember it. After completing of meiosis 1, another preparatory phase is present for a short period. Okay. In between, the preparatory phase which present in between meiosis 1 and meiosis 2, is called interkinesis. So after completing of meiosis, we can get 4 daughter cells from a single mother cell. Okay.